for my part, I would like to explain about the overall idea of our mini project. The objectives are prepare plant culture media, secondly, prepare expanse for tissue culture, thirdly, initiate in vitro culture of plant species, and lastly, raise aseptic seedlings for micropropagation. In vitro tissue growth requires tailored nutrients and a growth medium. Auxin and cytokinin concentration determine tissue culture initiation and development. Explants should be carefully chosen and sterilization must be done without harming plant tissues. Micropropagation involves initiation, multiplication, rooting of pre-plant and acclimatization stages. The initiation stage establishes aseptic cultures by placing disinfested explants on a medium. In the multiplication stage, shoot cultures are divided and recultured, or shoot formation is induced using cytokinins. The rooting or pre-plant stage encourages adventitious root formation using auxins. Acclimatization involves gradually transferring root shoots to soil while adjusting humidity to prevent wilting. Tomato seeds are used in this project for tissue culture.
Methods of preparing media preparation: 4 ml of macronutrients, 2.5 ml of micronutrients, 4 ml of vitamin stock, 2.5 ml of iron EDTA were added into a conical flask, and then the 400 ml of distilled water were added with the 15 gram of sugar, sucrose, and placed on the stir plate to mix. Then the pH was adjusted between 5.4 to 5.8, and they were divided into 2 flasks with a measurement of 400 ml in each by adding distilled water. Then BAP and NAA with the 4 gram of agar were added separately, and then they were poured into the glasses and autoclaved before keeping in storage. Next, sterilization of seed. Place the dried seeds in the conical flask and wash them with tap water 3 times and distilled water 1 time. Then they were placed in the lamina flow and converted into a sterile conical flask. They were added 5% of sodium hydrochloride and 3 drops of TN20 and swelled for 5 minutes. They were washed with sterile distilled water until the foam is not formed. Then they added 70% of ethanol and swirled for 1 minute and washed with sterile water again, and they were put on the empty dried petri dish and placed it on the medium for the further process, and they were kept in the growth room with the labels. Next, subculturing. After a few weeks of observation, the plants grown without contamination were selected to do subculture in a laminar flow. Used forceps that has been dipped in 70% alcohol were used to remove the plant culture from its jar and place it on the alcohol wiped plate. They were trimmed the explant, removing unnecessary shoots and roots, and separate them into individual plantlets. They were placed in a new medium and in a growth room for the further growth. As the plants grew well, they were acclimatized in the nature environment with some procedures.
We'll be discussing these 3 stages of tissue culture with different concentrations of BAP and the observations made during each stage. In the first stage, we successfully developed callus using BAP at a concentration of 0.5 mg per liter. Callus formation is a crucial step in tissue culture as it serves as the foundation for subsequent stages. The addition of BAP at 0.5 mg per liter stimulated the proliferation of undifferentiated cells and initiated the formation of callus. The callus exhibited healthy growth, displaying distinct textures and colors, which indicated its viability and potential for further development. Moving on to the second stage with BAP at 0.5 mg per liter, we observed the remarkable growth in shoots, leaves and roots. The culture thrived as shoots underwent active proliferation and leaves matured beautifully. The establishment of a well-developed root system was also observed, which plays a vital role in nutrient absorption and encourage the balanced growth between shoots and roots ensured the overall health and vigor of the plantlets, preparing them for their transition to ex vitro conditions. However, in this third stage, When the concentration of BAP was increased to 1.0 mg per liter, we encountered a different outcome. Callus formation was hindered, and the higher concentration of BAP exhibited adverse effects on shoots, leaves, and root development. We observed the absence of shoot elongation, underdeveloped leaves, and the failure of root formation. Additionally, contamination became a concern in the third stage. Fungal and bacterial growth was observed, which negatively impacted the plantlet's growth and development. Contamination spread throughout the cultures, leading to stunted growth, chlorosis, and disrupted organ differentiation. So maintaining sterile condition and implementing proper sterilization techniques are critical to avoid such issues.
Impacting the success of the outcomes of a mini project, which are culture medium composition and formulation, growth regulators such as BAP, sterilization techniques, incubation conditions, subculture and maintenance, observation and data collection. The experiment mainly focused on the impact of 2 mgl BAP on tomato regeneration. The results obtained from the BAP treatment are increased shoot length compared to untreated samples, more leaf developed in BAP treated samples, enhanced root development in BAP treated samples, BAP treated explants add more shoot buds, leading to increased shoot formation. There are 3 stages for culture medium supplemented with BAP at different concentration of 0.5 mgl and 1.0 mgl. Callus formation began as cell divided and clustered around the edges of the explants. The callus tissue grew and expanded, initially appearing pale or whitish, potentially developing a slightly greenish tint later on. However, there were no visible signs of shoot or root formation during this stage, indicating that the callus remained undifferentiated. In stage 2, shoots elongated and emerged from the callus tissue, while leaf primordia developed and expanded. Root primordia were initiated and elongated, leading to the development of characteristic shoot, leaf, and root structures, with shoots exhibiting apial dominance. Last stage of our tomato seed culturing, active shoot proliferation occurred, resulting in the generation of multiple shoots. Leaves grew and matured during this stage. The root system elongated, branched out, and established a network, maintaining a balanced growth pattern with shoots. Plantlets also underwent hardening of shoots and roots, preparing them for transplantation. 1.0 mgl BAP concentration resulted in the absence or slower formation of callus and shoots, inhibited leaf growth, and a failure to grow upper. Additionally, fungal growth appeared and spread, causing contamination that inhibited plant growth, disrupted differentiation, and led to distorted, abnormal, or absent shoots, leaves, and roots in severely contaminated cultures.
In conclusion, lower concentration, which is 0.5 mg per liter of BAP, resulted in successful plant development, including shoot multiplication, elongation, and root formation. And higher concentration, which is 1.0 mg per liter of BAP, led to contamination issues and inhibited callus formation, shoot and leaf development, and organ differentiation. For recommendations, to prevent contamination, strict sterilization protocols and aseptic conditions should be maintained during tissue culture. Furthermore, quality control measures are important, including regular monitoring for contamination, assessing plantlet health, and conducting careful observations and measurements for accurate results.
So that's all from me. Thank you.